today's speaker is Lars Fritsche, and he is Associate Research Scientist in Biostatistics.
Wonderful. So thanks so much for giving me the opportunity to present some of my tools here, and thanks for the kind introduction. So today I'm going to talk about PRS Web and basically about how we constructed, evaluated, and shared our cancer-related polygenic risk scores. And so the outline of my talk will be that I first will talk a little bit about polygenic risk scores and why they're useful to translate findings from GWAS into kind of a clinical setup maybe. Then spend time on how to construct polygenic risk scores and show some examples. And finally, show how we evaluated those cancer-related PRS in 2 large biobanks. And finally, show the platform itself that we use to kind of share the polygenic risk scores, but also share their performance and all other kind of features, so other people can download and try those risk scores in their own data sets. Okay, so first the clinical translation of GWAS findings. So the goal is here, we want to improve measures of disease susceptibility. And usually the classical way is genome-wide association studies that identify susceptibility variants. And so by doing so, they often end up in an interesting gene telling them about pathways. And so they get novel biological insights. And that is helpful because it points people into a pathway that might be targeted through therapeutics, might identify biomarkers that could be relevant for the disease diagnostics, but then also maybe there are some inferences you can make on how to prevent the disease. And here, the other half, how we're going to translate GWAS findings is to kind of improve how we measure the causal mechanisms or how we measure the susceptibility. And the idea is to have a kind of a personalized medicine that we can help to better diagnose an individual, so to support a clinical diagnosis. Maybe we can make some inferences about the disease prognosis or onset, and maybe we can use those data points to optimize the therapy. And this slide is from 2008, but kind of those goals are still up to date. And GWAS are very successful. And here I'm showing a plot from Mills et al. They kind of summarized GWAS that were published between 2007 and 2017. And what you can see here is that GWAS became more and more frequent. So more GWAS studies were performed. When you look at the color code here, blue is the largest GWAS category. You can see that sample sizes increased overall. And because of the larger sample sizes, then of course the number of association signals increased also with the increasing sample size here. So here we're looking at, I think there are 40 million analyzed individuals, and there are up to 20,000 associations that were discovered by 2017. And there's a market for those GWAS studies. It was still in 2017. There were more and more journals publishing those GWAS findings, and more diseases were analyzed. So this is a very successful approach. But when we look at the kind of the effect sizes and number of loci, we can see that in the beginning, people were picking up those low-hanging fruits, common variants with strong effects, but over time, those were already identified and became more and more difficult to find novel hits. But with the more samples they had in their study, they could pick up additional loci, but the effect sizes itself, they became smaller and smaller. Also, they're now able to also pick up lower frequency variants. And so there's not a linear trend on how much of the heritability can be explained, but it's more kind of, you can see here that they tend to reach a kind of a plateau. But still, even if the effect sizes are not that strong, and each single variant can explain only a very small part of the susceptibility of a disease, overall, the number of loci itself, they explain more and more of the heritability. And so we want to capture this heritability to make kind of risk predictions or offer risk stratification. And so the way we are going to do this is we're condensing the findings from a GWAS into a single biomarker. And so this is shown here. This is an example GWAS from the UK Biobank. Many hits here that reach genome-wide significance here in blue. And we calculate a single score based on those risk variants. And later I'm going to show how we're going to do that. And we are going to use this to differentiate cases from controls here, cases in black, controls in black, cases in red. And you can see there is a shift in the risk score distribution, and we can use that for risk stratification, for example. And so before I talk about PRS, I want to kind of set the expectations. So right now, the polygenic risk scores we're using, they're most useful in the tails of the distribution, and that can be shown here. When you look at cases and controls, you can see this area where there's a large overlap. So you're not able to differentiate cases from controls. But when you look at the tail here, you can see there's an enrichment of cases in here. And that's also shown here by the odds ratios. When you compare the top 25 versus the bottom 75, we get an odds ratio of 3. In this case, this is prostate cancer, meaning that those PRS allow you to identify a subset of a population that is at higher risk for developing a disease. Another thing to keep in mind is we're looking at genetic data. And so we have the different effects in population, bottleneck effects. And so many of those polygenic risk scores are ancestry specific. So the distributions differ between ancestry groups. And you need to be very careful when you have a polygenic risk score, because it's not directly transferable to a different ancestry group. And the problem in GWAS studies right now that we're still facing is the lack of diversity. There's a European bias in here, and later on I'm going to show a plot about that. Most of the GWAS that were performed are focused on European ancestry individuals. Another important thing when we talk about PRS is that they only capture the heritable fraction that is captured by the additive effects of SNP variants. And so this is kind of the maximum that we can predict with the polygenic risk score. And so what I'm showing here, these are different traits, and depending on the trait, there's a different estimated familiar heritability. This is the lighter color, and in the darker color indicates the heritability that we currently can explain by SNPs, by common SNPs from GWAS studies. And so there's still a gap in here, but the maximum here indicates that there's a proportion that is due to other demographic environmental risk factors that are not captured in polygenic risk score. And so this means that when you fully want to understand the risk for a disease, we also need to consider those non-genetic risk factors and current exposures that influence the susceptibility. Okay, so coming now to the PRS construction itself. There's a basic recipe how we construct polygenic risk scores. And here I'm talking about polygenic risk score because I mainly focus on case control studies. If you have quantitative traits, you would call them polygenic scores. And so basic recipe is the same. You want to select kind of independent variants that you think contribute to diseases independently, maybe different loci. And each one of them has a certain effect size or weighted effect size. Let's call it beta hat for a single SNP. And they're coming from an external GWAS. And here I'm looking at summary statistics. And then in your own cohort, you have genotype data. And per individual, you have either 2, 1, or no risk allele. And so basically you have a list of those risk SNPs and their genotypes. And now you multiply for each single risk SNP, the risk allele dosages times the effect size from the external GWAS, and then basically sum up those numbers into a single score, and that's what we call a PRS. And there are different ways how to weigh the effect sizes and how to condense those lists. And so the question is always, how do we choose the list? Which SNPs should be included in a polygenic risk score, and how many? And should we do anything with the effect sizes? Should we shrink them? And there are many different approaches by now. And here's a recent review article by Ying Ma and Zhang Zhou in our department, Yingma is actually a GSRA who's working with me on polygenic risk score, and she put together this very nice review manuscript, review paper. And here showing kind of the history of current polygenic score methods. And here in blue, these are based on individual level data. But I'm mainly talking about methods that use GWAS summary statistics. They are shown in orange. And you can see this is a real trend here. More and more people are developing methods, including additional information, improving the ways how they weigh those effect sizes and simulate effect size distributions, and also include additional annotations from other sources. And so here, for our study, we focused on 4, and actually there's the first method actually is the simplest one, where you just focus on the independent GWAS hits. And that's, I think, the most common polygenic risk score method itself. And then there's LD clumping and thresholding, LD pred, lasso sum, and PRS CS auto, and I'm going to talk about them in the next slide. So you always have the kind of, you're faced with the choice that you have to make, and here we said we focus on those 5 because we can make them work on our resources we have, and they work quite well. Okay. So focusing on those 5 methods here, the simplest one is the fixed p-value threshold, and that is, for example, the GWAS hits. And the kind of you want to ensure that they are independent, so you perform an LD clumping. What it means is you take the strongest signal, and then you determine which of the other SNPs are correlated to that one. You exclude those, and then go on to the next one that is then independent from the first you selected, and you do that until you run out of, for example, SNPs that reach genome-wide significance. So that's the most simplest approach. But then there's an advanced method that says, oh, the genome-wide significant threshold, maybe that's not the optimal threshold we want to use. We are talking about prediction, so it might be helpful to go to pick SNPs that did not reach a threshold. So we might increase the risk of adding noise to this risk score, but still there are informative SNPs in here that could contribute to the predictive power. And so we don't know this threshold, so there's kind of a tuning step involved where you determine this optimal threshold for this score. And one example, one tool is called Precise2 that does exactly that. There are other approaches, penalized regression approaches, that try to balance bias and variance to find optimal parameters. And they also they adjust for LD. This is part of their algorithm. One example for that would be lasso sum. So this more advanced method, you can end up with millions of SNPs that contribute to a risk score, or you can use a Bayesian method, where you kind of use some prior information or assumptions. Let's say you assume a certain percentage of variants that contribute, that are causal, and so you model that fraction into a risk score, and also you shrink effect sizes with respect to LD, and then you have, for example, PRS CS auto, one SNP that usually includes over 1 million variants, or you can use LD pred, and depending on the fraction of causal variants, you can end up with very few contributing SNPs or many, depending on the assumption of that approach. So basically, you end up with, depending on the method, you end up with very different SNP lists. And so when we want to construct PRS, we aim to have at least 3 independent data sets. So we need the discovered GWAS studies that we use to find susceptibility variants. And then we need a training set that we use to kind of get the tuning parameters that tells us this might be the setting that works best for that GWAS and that phenotype we're interested in. And because you don't want to avoid overfitting, you want to evaluate that PRS in a testing set. And you can also think about a 4th data set where you then actually are going to work with the PRS, but here I'm focusing only on those 3 independent data sets. Okay. And so the 2 data sets we are going to use are 2 biobanks. One is the Michigan Genomics Initiative from Precision Health here from the university, and it's a hospital-based study, and you might have heard about this from a previous presentation here. And so right now there are over 80,000 participants, maybe more closer to 100,000 by now. And so the kind of main recruitment mechanism was through surgery or other interventional diagnostic procedures, and also people consent, and then they give a blood sample, and we can access their full electronic health record and be identified. And the catchment population of Michigan Medicine is mainly European ancestry, and so also in this data set here, we end up with over 92% European ancestry individuals. The data set is a genotype based, so genotype array based, but then we perform imputation. And here I'm using the haplotype reference consortium data set. But the latest data set is the TopMed imputed data set with, I think, right now with over 50 million SNPs that were imputed. In contrast, the UK Biobank study you probably all know very well includes up to 500,000 individuals, and over 95% of them are of European ancestry. And here again, I'm using a similar imputed data set, the HRC imputed data set with over 20 million variants. So we have the genetic data, and we have electronic medical records that we can use to construct phenotypes. So this is an ideal setting to generate many different phenotypes. And because I said in the beginning that polygenic risk scores are ancestry specific, we are focusing on European ancestry. That makes the whole analysis easier because they are mainly European GWAS available, and we need kind of the matching ancestry when we construct PRS. And so right now we are limited by diversity, and so again focusing on European ancestry. And this leaves us with close to 40,000 individuals in MGI that are unrelated, and 410,000 in the UK Biobank. The percentage of females is similar, and they have a similar mean age, but the age distribution is slightly different here. But for both, we have rich diagnosis data. And here I'm looking at ICD-9 codes. So these are international classifications of disease codes, and they help usually with billing or for administrative purposes in the clinic. So they're not ideal to define phenotypes, but they can be used to define phenotypes, and I'm going to show that later on. So we're going to use those electronic health records, in this case ICD codes, to define case control studies. And the most convenient way to do this that we found is the so-called GWAS code system, and you might have heard about this. The idea here that was published by Josh Denny's group in 2013. I don't know how to do this. Oh, here we go. There's a GWAS catalog, basically tells you which ICD codes can be collapsed into a broader phi code term. And the way it works is that you have ICD codes for a diagnosis that describe the same phenotype, and you would use those codes to define cases. You have then other diagnoses that might describe a similar or related phenotype but does not match the phenotype, so you want to exclude those. And all the other diagnoses you consider as controls. So even if you're looking at a hospital-based studies where you probably don't have healthy individuals because there's a reason why they came to the hospital, you can use them as controls. And by doing so, you end up with case control studies. You have a phi code per case control study. And it's really fast, this approach. These are not the ideal phenotypes if you're focusing on one specific disease. It's definitely helpful to work with domain experts to hone the phenotype definitions and get deep phenotypes there. Okay, so when we do this with the two data sets, we ended up with almost 1,700 phi codes that had at least 50 cases in MGI, and we had over 1,400 phi codes in the UK Biobank that had at least 50 cases. And when the collection of phi codes, in this case, this is what we call a phenome. This will be relevant later on when I describe a phenome-wide association study. Basically, we can use those 1,500 phenotypes and do associations with each one of them and get kind of an idea how specific a polygenic risk score is. Okay, so when we looked at the MGI study, and this is the main focus, main study I'm focusing on in my talk. we found that it is highly enriched for cancer patients. There are multiple reasons for that. As I said, the recruitment mechanism is surgery, and apparently cancer patients undergo surgery more often. Maybe they have biopsies. And also we have experts here in Michigan medicine, for example, focusing on melanoma. So the recruitment here is definitely enriched for those patients. MGI is much smaller than Leuka Biobank, but here when you look at the different case counts in MGI, they are sometimes comparable. Sometimes they can't reach the numbers that are observed in Leuka Biobank. So the sample sizes are quite different, but both biobanks have sufficient sample sizes to be used for the PRS construction. Because they are enriched for cancer, and we are especially interested in cancer, being affiliated with the Royal Cancer Center, that's the first category of traits we explored here. Okay. So coming next to the PRS evaluation, as I said, we have 3 independent data sets, and there are different setups you can imagine. So you can use the Leuka Biobank as the training set, and use MGI as testing, or vice versa, MGI as training and Leuka Biobank as testing. But then the question is, where do you get the discovery GWAS? And we found that it's specifically hard to find those summary statistics online that are freely available. And so there are some consortia that are very open about sharing their GWAS summary statistics, and breast cancer and prostate cancer are 2 of those cancer consortium that are fantastic in this regard. But we found that if you want to do this pan-cancer across many different cancers, you often don't find those. So what we did now, we decided to use the Leuka Biobank also as our discovery data set, and split our data set in half. Use 1 half for training and 1 as testing set. And so this way allowed us to generate many more PRS for other cancers that are not openly available online. Still, there are large GWAS that are available, and we can use them to generate PRS also in Leuka Biobank. As I said before, here I'm mainly focusing on MGI. Okay. Of course, there's a difference in the quality of external GWAS. So if you have Leuka Biobank, you're limited by the sample size, and they nowadays can't compete with those large GWAS meta-analyses. So you can see here, this is the Leuka Biobank, and in this case, this is on prostate cancer. This is the practical consortium. You have 10-fold sample sizes, and of course, you can see they are able to identify many more variants. And so this large GWAS probably will result in the better PRS. But as I said before, not all of those GWAS summary statistics are openly available. Okay, so what we did, we kind of looked for resources online where we can find SNP sets or GWAS summary statistics that we can use to construct polygenic risk scores. One source was the GWAS Catalog, basically a resource that tries to collect all genome-wide significant hits. Sometimes they also go below that threshold. And so we could do this for, I think, 24 different cancer traits. We also looked online through PubMed if we can find summary statistics that were shared with the publication. We found 20 of those. And for the Leuka Biobank, we didn't have to run those GWAS by ourselves because other groups already just used all the data they could find and ran GWAS for every trait they could define. And also they used, for example, the PRS code system that was done by Sean Lee, Department of Biostatistics here, or Ben Neal's lab, and they just put them online and you can download those GWAS summary statistics. And so in total, we ended up with over 200 SNP sets that we manually mapped to over close to 70 different cancer fee codes. And so how we used those different sources of GWAS summary statistics to test the different methods I mentioned before. So we did LD pruning and clumping, did the p-value thresholding. So this is the CNC approach, the fixed p-value threshold, for example, the GWAS hits, and then the more complex method, for example, lasso sum on PRS on the data sets where we had full GWAS summary statistics. And then we evaluated the PRS in terms of overall performance. Could be, and that's one way to do this is looking at pseudo R square. We can look at the discrimination between cases and controls. This is the area under the receiver operator curve. Or we can look at accuracy, and that could be the Breyer score. Also, we're interested in association strength itself. Is this PRS associated with the trait we're interested in or not? And then we want to see if this PRS is able to enrich cases in the tails of the distribution. Okay. As you can imagine, we generated many PRS while doing when we combined the number of SNP sets by the different PRS methods. And also we had 2 biobanks, so we ended up with over 1,300 PRS that we constructed. So the questions are, of course, what is the PRS method? What's the best external GWAS source? And is the method GWAS combination, is this one best solution that works for all the different applications, or is it dependent if you want to look at risk stratification, at association, or discrimination? And so those are the questions that we had. And so the metrics that we used are initially the association of the continuous PRS with the trait of interest, because those methods, even if we have all the data, they don't work. So you end up with a PRS that is not associated at all. Maybe the sample size was not sufficient from the original discovery GWAS, maybe sample size is not sufficient in our data set. And so we kind of use this initial filter here to make sure that we only analyze PRS that are positively associated with the trait of interest and at least nominally significant. And that directly, I think, cut the number of PRS in half. Then we looked at overall performance. That's the pseudo R-square, that's the measure that we use to rank the tuning parameters. So the one with the highest pseudo R-square, and that's the tuning parameter that we use then for the evaluation later on. Then we looked at in the testing data set, we looked at discrimination, here covariate adjusted AUC, looked at accuracy, and then also looked at the ability to enrich cases in 5 selected top percentiles, and this was top 1, 2, 5, 10, or 25%. Okay, so we generated a lot of data, and we ended up with over 500 PRS, over 30 cancers in MGI, over 20 in MGI. Okay, and here I want to show you some examples how you now can compare those polygenic risk scores you generated. And so one question is, what method works best? And so you look at the same GWAS source and look at the performance across those different methods. And here what I'm showing here, this is PRS, this is the Bayesian method. Plus with some, this is the regression method, penalty regression, and you have clumping, thresholding, and then different fixed thresholds. And I already ordered here by decreasing number of SNPs. So this Bayesian method includes over 1 million SNPs condensed into one single score. Plus some trimmed this number to 100,000. But then depending on those thresholds, you in the highest setting, you end up with 264 SNPs. And what we found here, and I already ordered it by performance in terms of pseudo R-square, that PRS was the method that worked best. And one of the reasons that we also found PRS work very well for many other traits. That is, PRS seems to work very well with different kinds of genetic architectures and adapt to the phenotype and GWAS summary statistics, and usually that helps with the performance. Also, PRS had the highest accuracy. So here, the smaller the better, and the discrimination was also better than the others. And the association strength itself, so the continuous PRS. So here, this is per one standard deviation also was the highest in here. Then when you look at the enrichment, in this case, top 1 versus bottom 99%, here you found that actually one of the other methods, the fixed threshold at 5 times 10 to the power minus 7, that performs best. Usually this method here only includes kind of the strong top signals here, and apparently that helps to define people that are at high risk. And that might be one of the reasons why this method is better for enrichment analyses. But you look at 1%. This is not always as clear cut. If you look at prostate cancer here, you can see that discrimination, clumping, and thresholding works best. For association, PRS works better. But then when you look at the enrichment in top 1 versus bottom 99%, then in this case, Lasosum works best. So there's not one solution that works best for every single approach here. Then, of course, we're interested to see which of the GWAS sources works best. We had different sets, for example, a large published study on breast cancer. We had used all the top GWAS hits from GWAS Catalog and used 4 different GWAS on MGI Biobank breast cancer cohorts. And here they use different phenotype definitions, so that's why they are different. We found that this large GWAS meta-analysis with the largest sample size that outperformed all the other approaches. And that's what we usually see. The latest, largest GWAS, this is the best source you want to have to construct polygenic scores. But often those are not available, so you're limited to this down here. Again, here, the large GWAS perform best across all different metrics we have. Okay, so when we look at discrimination now across cancers, what we found is that depending on the cancer trait, this discrimination ranges from coin toss to 0.7 maybe here. And so for some traits and for some GWAS summary statistics, this works quite well. But when you're honest and you consider how you classify this, the accuracy, this is by definition a low discrimination, meaning that those PRS are not really helpful for individual level prediction of diseases. And so by themselves, I would say a PRS right now that we looked at, they are not useful to predict an outcome. But if you combine it with non-genetic risk factors, you might end up with much higher AUCs. And the reason for that is something that I've shown before. When you look at the distributions, there's a huge overlap between the two, so you can't differentiate a person just based on a PRS. Yeah, it's a case or control. When we look at the risk stratification, we found that for many cancers, we are able to enrich cases in the top 10 percentile. So here, for example, most extreme example is chronic lymphoid leukemia with estimates between 4 and 5-fold enrichment. Prostate cancer follows by 3-fold enrichment in the top 10%. And if you look at top 1%, those numbers increase as well. And so we created all those PRS, we did all those analyses, and one dilemma that we had is now we have all those results, how should we put them in one manuscript, how we can share them with the community. And so we came up with an online solution, and that's the last part of my talk, talking about the PRS web, how we shared them online. And here I was fortunate to work with Snehal Patil. You might know her, very talented application programmer, and it was kind of fantastic to directly work with her one-on-one to kind of discuss what we have, what we can share, and how we can make a user experience better to share the information that we had. Okay, and so basically now I'm sharing the platform itself, and we call this platform Cancer PRS Web. And so this is the first instance of PRS Web. We plan to expand this for other categories in the future, but as for now, we focus on cancer traits because we have this huge enrichment in MGI. And when you Google PRS Web as one word, you usually find this as the top hit here. In here, you can find the evaluation of PRS, the PRS evaluation table by phenotype. And then also we share phenome-wide association studies results. So using this PRS to screen for associations across the phenomes that we have, and then also share the file with all the information you need to regenerate this PRS in your own cohort. And so these are live examples. If you click on one, you kind of see what's going on there. And the key entry point here is the selection menu, where you select the cancer trait, your evaluation cohort, and then, for example, the enrichment you're looking for, or the risk stratification. And so nice feature here is you don't have to scroll through all those 68 cancers that we analyzed, but you can just type a word and it will autocomplete here and show you selection here, and also warn you if there's a phenotype where we're not able to find a PRS that we consider worthwhile analyzing. And here, when you type in lymph, you end up with those different phenotype codes we had PRS for. And here I'm selecting chronic lymphoid leukemia. Next, you can select if you want to look at the MGI Biobank or MGI. And finally, you're interested to compare the top 1 versus bottom 99%, or you can look at the others here. The reason why we did this last part here is because the table would get too broad if you include all the different tables that you're looking at. Then you select Show Table, and this is what you see here. In this case, we generated 3 PRS for chronic lymphoid leukemia, and you can see the GWAS sources, GWAS Catalog, and then 2 MGI Biobank GWAS datasets we used. There's information about the method, how many SNPs were used, and this is sometimes helpful if you want to avoid you working with a PRS that includes 1 million SNP, and there's another one that only includes 100 and performs almost as good, then you probably might opt for the other one. You get the association in the testing set, the other metrics. And so this is an interactive table. You can sort by your favorite metric and then select the PRS for the next step. And so also we excluded some PRS, those that did not perform well, did not reach nominal significant, or actually pointed in the wrong direction. We try to put as many warning signs as we can on here. And as I said, this is interactive. And maybe I get a chance later on to maybe show this platform. Snehal did a fantastic job, and this works very smoothly. So I'm very impressed by that. You can download PRS from this table here. And in here, you will find all the information that you need in a header, and then the chromosome position, and then the effect allele, and the non-effect allele, and the weight that was used to construct this PRS. So that's basically the recipe you need to reconstruct it. And then there's a neat feature in here that we kind of did not make very obvious here, but there's this PRS fibers column here where you can look at PRS fibers. And when you click on that, first you get a header in here describing the PRS itself, where it's coming from, how it was defined, which method, and so on. And then you can look at this fibers plot itself, just the Manhattan plot, kind of a Manhattan plot, but here you're not looking at SNPs on the x-axis, but actually at, in this case, I think over 1,600 phenotypes. And you're now associating each of those case controls that you're testing the association with the PRS that you have. And this is a nice way to kind of get an idea how specific your PRS is. Can it pick up the trait you're interested in? And then also sometimes you end up with secondary signals. And at this point, we are not sure if this is a precancerous condition. Maybe that's helpful for predicting the cancer outcome. Maybe it's a consequence of the treatment. And so that's something that we struggled in. This is all timestamp data, but here we're looking at case control studies without this time information. So we don't know if it came before or after this diagnosis. And so the way we resolve this is through, and those plots are interactive. You can hover over those dots and you get information. And Peter, actually in the audience, he is partly responsible for this nice interactive locus zoom feature that we're using. Okay, so if you want to exclude, you want to know if these are independent associations with the PRS, you can exclude all the cancer cases and see if those associations remain. And that would be an indicator if that's an independent association with a shared genetic component. And so we also offer those exclusion PRS fibers plots. And of course, people are sometimes, so I'm a very visual person, but if you're more interested in the raw data and in the tables, you can look at table view and you can see the PRS fibers and the exclusion PRS fibers side by side, effect sizes, number of cases and controls included in here, and you can download this also as an Excel file. Okay. There's a lot of information in those tables, and sometimes you want to know how was the phenotype defined, what was the method that was used. And so we tried to incorporate that information in those tabs in here. So we have methods here. This is basically the method sections that we had in our manuscript. You can read about the different cohorts that we used. Then you might be interested in getting an overview about all the PRS that were generated. You can see the different traits in here and this description and the fee codes. You can see how many remained after filtering, how many PRS were there before we started, and you can look at PRS MGI or MGI Biobank. So this is a nice overview. And if you want to see how a phenotype was defined, you can click on a fee code and it will tell you those ICD codes were used to define in this case, the prostate cancer diagnosis. Okay. So, okay, and at this point, I might be able to give you a quick live demo. I've shown you most of the features, and so now I have to switch. Supposed to go here. Here we go. Is this available online? Yes. So here, as I said, the key here is to look at the selection menu that is not showing up. Here we go. So one nice example here that I know works well is, I think, basal cell carcinoma. And we want to look at this in the UK Biobank, let's say 5% versus rest. And so we want to show this, look at this table. And you can see the selection was very smooth. You could just type in a word and the phenotype was showed up. And here we can see that there are two studies that went into this score, 24, 28 SNPs. The association was extremely strong. POC was lower as with our other yes, we had in our data set, and still this PRS is able to enrich cases in the top 5%. And when we look at the PRS GWAS, the data is loaded in here, you get the details, and you can see skin cancer, melanoma, and carcinoma are showing up on top here. So this seems to be a skin cancer related PRS with many associations across different types of skin cancer. And the reason for that is that I think pigmentation itself, the genetic factors that influence pigmentation are causing skin or strong risk factors for skin cancer, and so they're relevant for all kinds of skin cancers here. But also you can see that there are dermatological traits in here that seem to be associated. And so when you look at the exclusion PRS GWAS, in this case, you can see that here there is a trait called sebaceous cyst that is associated with this PRS also in the exclusion PRS GWAS, meaning that this trait itself seems to have a shared genetic component. And, here, likely coming from the pigmentation again. So when we select this in here, you can also directly see the association of the continuous PRS. And you can see Q1 versus Q2. You can see if this enrichment here is present in the different quartiles. And in this case, it is present in the PRS, but also in the exclusion PRS GWAS, meaning that there seems to be an independent association there. If you want to go at the table, you look at the table. This generates here automatically and then sorts by p-value. There are some extremely small p-values that shows up as 0. You need to fix that. But again, you can look at all those tables and sort them as you want, depending on your interests. And I think, please go to PRS Web, try, explore, and if you find any bugs like those 0 p-values, let me know. Can certainly fix them. Okay, so what's next? So we want to expand our platform to other phenotype domains. We started with cancer, but we have a whole phenome, so we might be able to do this across the whole phenome. Right now, we are focusing on exposure PRS, so not only at case control GWAS, but on kind of traits that are relevant for other common diseases. Think about lipid levels or BMI levels. They also can be useful as predictors for common diseases, or you can use them as proxies in case you don't have them measured in your own data set. And that's why we think exposure PRSs are relevant for the community. We are using MGI, and so many researchers are by now using MGI, and so we want to return those PRS we generated. Sometimes it takes a long time to condense 1 million SNPs into one score. And so we have those numbers, we can directly share them with Precision Health, and so they can make them available for other researchers. Of course, we want to try new PRS methods, and ideally we want to increase diversity, offer also non-European PRS. But here study from 2019 shows that when you look at the GWAS catalog, almost 80% of the individuals that are analyzed are of European ancestry, followed by East Asian. There are some African cohort in here, and some cross ancestry studies are in here as well. But kind of people have to do a lot of catching up to increase diversity for GWAS studies. So our work is published in the American Journal of Human Genetics a year ago. And again, I want to especially thank Snehal Patil with all of her patience to put together this webpage and kind of discuss what is possible, what is not possible. It's always difficult to know, but she did a wonderful job there. And this work was co-led by Brahma Mukherjee, fantastic friend and collaborator. And finally, I want to acknowledge all the study participants in MGI and the UK Biobank. And then we worked with many different centers here at the university and departments. And we had some funding from NSF, NIH, and also through Precision Health from the university. Thank you for your attention, and happy to take any questions you might have.
Anyone in the room has a question? Feel free to ask a question. If anyone online has a question, you can either put it in chat or use the Zoom controls to raise your hand and I can call on you.
Yeah.
Two questions. One is more UI focus and one is more on the solution focus.
Okay.
For the first one with the UI for figure 2, when you were earlier when you were giving the live demo, is there a way to zoom in so you can see some of the different traits that so if you go to the graph option, yeah.
Oh, so this is.
I noticed that the figure 2 when you have the graph and table option, it was showing some of the different.
Maybe it was this one here.
That.
Sure.
I did not think that much to but it was with the number.
Not exactly sure which page you're referring to.
You had a selection of multiple changes and you showed on the graph and you could click and then you can take a look at the top 25, bottom 25, and then.
Oh, okay.
Yeah.
So that is this option here.
But my question was it possible to zoom in further on the map in case we see some of the data points being squished on top of each other?
Oh, I see.
So can we repeat the question by chance? The people online or seem to be unable to hear the questions. If you can speak up a little bit more, or Lars, if you're able to repeat, that would be helpful.
Sorry, I was asking for the graph that was generated. Is it possible to zoom in on the y-axis in case
Oh,
you
I see.
some of them all squished together?
Okay, so that is a good question. So we have this plot here, and sometimes there are some things happening, and you can see here there's a lot of overlap. Right now, I think those labels are, the script is trying to repel them, but still there's a lot of information in here. And I think at some point there was an option to make this to zoom in, but this is not an option at this point. But I think would be a very good idea.
Apparently, you can drag axis tick marks to zoom.
Oh, it's a new feature.
Andy wrote that in the chat.
Axis tick marks. Okay, maybe I'm doing it wrong. Here it is.
Yeah, you said like by the label, so like by 10, you could maybe drag the tick mark.
It's possible, but I disabled that. No.
Okay.
But yeah, that usually would be possible.
Yeah, okay. Might be an option. Yeah, but some of those plots get very crowded. Yeah, you're right. Which is a good thing because there are many interesting associations you can follow up on. But sometimes it's hard to see it. I think that's why we have this table option where you can look at the topics in the table itself. And you can directly see also what happens when you have an exclusion PRS. What are the traits that are removed when you exclude, in this case, skin cancer. And so these were all the skin cancer traits. What was the second question?
The second one was for the definition of the different demographic information for like Hispanic or Latin Do you distinguish that? Because I know that there can be some overlap with Hispanic for both one demographics or with the mixture with the American population. Or
I see. So here, this is not our study. This is a study that summarized the GWAS Catalog studies. And I think in here, there's kind of the curators of this database, they tried to categorize the ancestry groups. And I think these are the broad definitions in here. But you're right, there are better ways to do this. And for example, in MGI, we can use the inferred ancestry information from admixture or from PCA projection to better differentiate the different groups. And depending on which critical component you use, you can maybe differentiate between Hispanic and non-Hispanic individuals also. But as I said, the numbers are very low right now, so we can't offer it at this stage. But precision health is increasing diversity in MGI by specifically working with other communities that have higher diversity, and to especially increase numbers for Hispanic ethnicity or for African American individuals. Okay. Any other questions?
Not seeing anything else in the chat. We can just give it another moment just in case anyone's in the middle of typing, and then start again. Ryan said thanks, great talk.
Okay, thanks, Ryan.
Chris also said thank you.
It's live here.
All right, so I'm not seeing any more questions, so let's give our speaker one last round of applause. Thank you all for attending, and hopefully I'll see everyone next week.
Thank you very much.